pretty much associate that with 9331.
Dihybrid cross, 9331. And it makes sense that the dominant traits are going to show up more often, right? Because you only need one copy of that allele, that dominant allele, to see the dominant phenotype. So most of them are going to have the dominant traits. And that 1/16 is going to be homozygous recessive. That's why I said earlier, it's funny that the P's that I'm used to coming in contact with are the round, or the wrinkled and the green. But those are actually, if you took a normal P's and crossed it, you would only get that 1/16 of the time. So, other questions?
Why is in your first grain square, your little r and then your two big y's? That looks so weird. It would seem like it's got two dominant pieces to it. Why does it look so odd?
Well, so this is talking about the shape of the seed, right?
Yeah.
So round is dominant, or smooth, round, smooth, same thing. So wrinkled is a recessive. So this is going to be homozygous recessive for the wrinkled. So it's going to have a wrinkled phenotype. But it's got the dominant alleles for yellow. So it's going to be yellow, but be wrinkled. So this guy would fall into this 3/16 category. Just like the opposite of that, let's see. So this one is yellow but wrinkled. Where's our round but green?
That's important.
Yeah.
Other way.
Round and green.
Up.
Okay, hold on. I just gotta figure this out myself. Up. That's not telling you anything. So green and round. This is green and round.
No,
Two to your left.
two to your left.
That's also green and round.
It's
Yeah.
the exact opposite.
Okay, yes, it's the exact opposite of this one, but they're still both green and round. All right, anyways, yeah, so the opposite of that, you're going to show the dominant shape, which is round, but you're going to have the recessive color, which is green. So that would be this guy right here. So this is when you have one that's dominant and one's recessive, and the two combinations of that. And the other possibilities are all recessive or all dominant. Questions? I see some people nodding their head yes.
What's the difference between an allele and a gamete?
An allele is, let me back up. Your gamete is your sperm or your egg. It's that simple. Allele is just a version of a gene. So like the allele for brown hair versus the allele for blonde hair. The allele for blue eyes versus the allele for green eyes. So alleles are just different versions of genes. Other questions?
What is the difference between a phenotypic ratio? Well, yeah, what's the difference between a phenotypic and a genotypic?
Okay, so when we're looking at, let's go back to this example right here. So we'll go back to the brown hair. So the phenotype is what the individual looks like. So my phenotype for eye color is brown because I have brown eyes, just like my phenotype for hair color is brown because I have brown hair. So the phenotype is what it looks like.
Okay.
The genotype is what the actual different alleles are. Whether you have two dominant ones, two recessive ones, or one of each. That's the genotype. So in this example, if we crossed, again, a homozygous dominant male to a heterozygous female, we're going to end up with 100, no, that's not what I meant to do, but let's do this instead. All right, so the genotypic ratio, you look at the actual alleles, right? We've got 25% homozygous dominant. We've got 25% homozygous recessive, and then 50% are going to be heterozygous. So that's the genotypic ratio. That's what your genes are telling you.
Okay.
But the phenotype, because this and this, although the genotypes are different, they're going to have the same phenotype. They're still going to have brown hair. So the phenotypic ratio is actually what you see, what you're looking at. And the genotype, I don't know what my genotype is for my hair. I could be big B, little b, or I could be big B, big B.
Okay.
You see? So you can have a different genotype but the same phenotype. Does that make sense? Okay. And so the phenotype of this guy would be blonde hair,
Okay.
but his genotype would be homozygous recessive.
Oh, okay, that makes sense.
Okay.
So if you were to say, give me an example of a genotype, I would be able to say heterozygous.
Yeah.
Okay.
Yeah. All right, other questions? Okay, so we've already talked about Mendel's law of segregation. And again, all that's saying is that two versions of a gene are going to end up in different gametes. And that's just based on the physics of, or not physics, but the mechanism of meiosis. That's just always the way it is. Because you're separating homologous chromosomes and then you're separating sister chromatids. And because these are haploid, that means you're only going to have one version, right? One allele, not two. And so the other law that Mendel came up with or discovered is the law of independent assortment. So remember when we were talking about meiosis and all the different combinations or all the different ways that we can introduce genetic diversity? You know, one way we talked about was crossing over. And the other way we talked about was random fertilization, you know, random sperm, random egg. Well, the other one we talked about was the fact that it depends on how those homologous chromosomes line up at metaphase 1 of meiosis 1. And so this law that stems from this is called the law of independent assortment. Remember we said that this pair of homologous chromosomes is going to line up independently of this pair? That's what the law of independent assortment is. So the homologous chromosomes are going to assort themselves or line up however they want randomly. And how these line up is totally independent of how these line up. And so depending on whether you have, you know, mom's chromosome on top or dad's chromosome on top is going to depend on the gametes that you're going to get. So we have different possibilities right here. So in this first possibility, we have all of dad's chromosomes on the top and all of mom's on the bottoms. So whenever meiosis 1 occurs, we separate the homologous chromosomes. Now this has all of dad's DNA and this has all of mom's DNA. So that's going to affect, you know, what the gametes look like. Now in this other possibility, we have, you know, one of dad's on top and one of dad's on bottom and one of mom's on top and one of mom's on bottom. So meiosis 1, again, we're separating the homologous chromosomes, but now we have one full chromosome from mom and one full chromosome from dad. And so again, after meiosis 2, that's going to change the pattern of your alleles. So the law of independent assortment, all it's saying is depending on how these homologous chromosomes line up during metaphase 1 is going to determine what combination of alleles you're going to get in your gametes or in your offspring. So it's something we've already talked about. I just didn't tell you it was a law until today. So the law of segregation says that two alleles for the same gene after meiosis are going to be in two totally different gametes. The law of independent assortment says that depending on how those homologous chromosomes line up during metaphase 1, that's going to dictate what those gametes are going to look like. And so this was an example showing you the different possibilities. And remember, I gave you the stat. In this example, there's only, you know, 2 traits that we're looking at. But for humans, you know, we have 23 chromosomes. So there's 2 different ways that each pair can line up, right? Either with mom on the left or dad on the left. So we have 8.3 million different possibilities of how those homologous chromosomes line up. So if you got that many different possibilities, that means you're going to have 8.3 million different types of gametes. So lots and lots of genetic diversity. All right, y'all okay with independent assortment? Is that a no or a yes? Maybe a halfway? Okay. So everything we've been talking about to this point has been what's called Mendelian genetics, you know, named for the scientist who discovered it, Gregor Mendel. And so Mendelian genetics is like the default type of genetics, or the default inheritance pattern. And so we actually have other types of inheritance patterns that are possible besides Mendelian. And so this chapter is focusing on different inheritance patterns. All right, Mendelian is one inheritance pattern. Now for the rest, we're going to talk about other different types of inheritance pattern. Another type of inheritance pattern is called incomplete dominance. So we're still going to have dominant and recessive alleles, but they're just not going to behave the same as they would in Mendelian genetics. So in incomplete dominance, what that means is you're going to have two alleles, and one of them isn't going to dominate the other. They have the same amount of strength, so to speak. So an example is going to be in snapdragons, which is a type of flower. So if you take a homozygous red snapdragon and cross it to a homozygous white snapdragon, you're going to get a combination of something in between. So you're going to end up with pink. And then if you cross pink and pink, you're going to end up with either red or white. So incomplete dominance just says that not one allele is dominant over the other. So red is not overpowering white, and white is not overpowering red. They're working together to make this intermediate phenotype. And so another way to say it is heterozygotes, which this guy is, he's got one copy of the red and one copy of the white. Heterozygotes are going to be halfway in between the parents when incomplete dominance is present. And hair is not one of those traits that is inherited incomplete dominance. That's why I said when you were talking about the sandy red and or sandy brown, sandy red, it's not a merging of phenotypes like this example is.
What about skin color?
Ah, we're getting there. Skin color.
I've been wondering that since we started this.
All right, we're getting there. We're getting there. I can spoil it now and tell you, or I can.
No, don't spoil it.
Spoil it.
So I know it.
Like
No.
a batch of running.
Hey, we lost you 30 minutes ago, so you just go ahead and tell us.
All right, we'll get there. We'll get there. That's another type of inheritance pattern.
Because I'm wondering, if I have a baby with another black person, that baby will be dark.
Yep. So, just like if you had a baby with a white person, it would be kind of somewhere in between, right? So color, like skin color, is based on, there's multiple different genes that are contributing to that. And it's all about the amount.
I don't want dark kids.
Really?
All right, let's not get into this discussion. Let's just stop that right now. Hold off on that. Hold off on that. All right, so are y'all okay with incomplete dominance?
I was just gonna ask, I think I jumped ahead, that's the same thing as codominance though, isn't it? Because you said if there's not one that overtakes another?
No, that's a little different than codominance.
Okay.
So in this case, we don't really have, we just have alleles. They're not dominant or recessive.
Okay.
In codominance, you're going to have two dominant alleles. But these aren't dominant or recessive. They're just kind of on their own, and they give you this intermediate phenotype. All right, so another type of dominance is called, or another type of inheritance, I should say, is called codominance. And by the way, when we're talking about this Mendelian, when you have a dominant trait overpowering a recessive trait, we call this complete dominance. So one allele is totally dominating the other. So these examples, all this we did over here, this is all complete dominance.
Is that considered Mendelian?
Yeah, it's part of Mendelian. Yeah, so in Mendelian and inheritance, you're going to have one trait totally dominating the other. So we call that complete dominance. And so these are kind of just different versions of dominance. You have incomplete dominance, where one of them isn't totally, they're not dominating one another, they're just mixing together. And then you can have codominance, is where you have two dominant alleles that are both contributing to the phenotype. Blood type is an example of this. So, you know, there's 4 different blood types, right? Or you can have BA, you can be B, you can be AB, or you can be type O. Now, the A allele, this is a dominant trait, or a dominant allele, but so is B. It's not like A is dominant, you know, it's not like there's more type A's than there are type B's because A is dominant and B is recessive. They're both dominant. It just depends on what you inherit from your parents. So,